So today I'm going to do a quick video for you on PhyloBook. PhyloBook is a tool to display and annotate phylogenetic trees, and it allows us to select and manually edit lineages. Just to give you a quick overview of what I'm going to talk about, I'm going to give a little bit of detail about what PhyloBook is, talk roughly how data sets are prepared for usage within PhyloBook, although we'll cover that in detail in a separate lecture. Talk about how the data sets are organized in PhyloBook, what files and data are contained within a data set in PhyloBook, and then we're going to talk about navigating, viewing, assigning lineages, and exporting data and lineage assignments from within PhyloBook. So PhyloBook is a tool that allows us to display and annotate phylogenetic trees, and we can share that data to collaborators through a password-protected website. And in this website, what we can do is we can set up users and groups of users, and then we can assign various data sets to belong either to specific users or specific groups of users. So this allows us to limit who sees different sets of data. If we have a collaborator that works on one project and a different collaborator works on a different project, we can limit the data so that each collaborator only sees the data associated with their project. One of the big strengths of PhyloBooks and what our primary use of PhyloBook is that it provides tools to identify lineages within a phylogenetic tree, and then we can manually edit those lineage assignments to improve them. Lineage assignments can be initially provided by some external tool like a clustering algorithm or any other algorithm that you have to automatically assign lineages, and then it can be manually edited. Manual editing or lineage assignment is aided by highlighter plots. Highlighter plots are plots that came from a concept initially created at LANL, or LANL, the Los Alamos National Lab. And what these plots are is these are plots that highlight mismatches between the sequences in the tree and a reference sequence. There are also match plots that will highlight matches between sequences and the assigned lineages, and the combination of these two plots make it relatively easy to determine if a given sequence is a recombinant between previous lineages, etc. Once we're happy with our lineage assignments, we can then export sequences in lineage-specific FASTA files for further downstream analysis. Our primary use for PhyloBook has been to look at HIV sequences in infected subjects who participated in clinical trials for vaccines or monoclonal antibodies. And because our interest is looking at selection on those HIV sequences by that treatment, and because we are concerned that the selection might be lineage specific, we're designed a system that allows us to more easily select lineages and then export them. PhyloBook is a published piece of software, so we wrote a paper on it, and it's available in Biotechniques with this link here. And then, of course, the software itself is freely available via GitHub. So a data set within PhyloBook is a collection of related sequences. In our example, it might be a set of sequences of HIV that were obtained from a single subject. Those sequences are processed to often collapse the identical sequences. So when we do deep sequencing in a patient, there may be multiple copies of the same sequence, and we don't want our phylogenetic tree to be really tall, displaying all kinds of identical sequences. So we collapse the sequences into sequences where each one is unique. When we do that collapsing, we actually annotate the sequence name with an underscore and the number of sequences that were collapsed into it, and PhyloBook later on will make use of that information. Once we've collapsed the sequences, we will align them. You could use any alignment tool you want to, as long as you provide the sequences to PhyloBook as an aligned FASTA file. We're going to do some sort of tree generation, and PhyloBook again will accept files from any tree building program provided the output is in Newick format. But we have a pipeline that you can use to generate these files. And then a highlighter plot gets produced, and again, this highlights mismatches between sequences in the data set and a reference sequence. And in our case, the way its system is set up, it uses the first sequence in your FASTA file as the reference sequence. Now, all of these steps you can do manually with whatever software you like, but we have a pipeline that will perform the steps above, and I'll speak about how that pipeline works in a separate video. This is just to give you a quick idea of what that pipeline does. We start out with some collection of related sequences, collapse those sequences, you get some pre-processed files out of that. Those sequences are then aligned and flow into our pipeline that generates the phylogenetic tree. We use fig tree to generate an image of the tree. We use highlighter to generate an image of the mismatches between sequences within the tree and a reference sequence, and then those files go to PhyloBook. You can also have some external software that takes your sequences and tries to make potential lineage assignments. We do that with a simple k-meteoids clustering program, but there are other methods that we're working on to do that, and PhyloBook is set up so that if you send it the right kind of text file, it will take whatever your initial lineage assignments are and display those within PhyloBook. Alright, so now let's take a look at how PhyloBook itself functions. When you first land in PhyloBook, at the initial landing page, there is a collection of projects, and within these collections of projects, we can even have a higher order folder. So in this case, we have a manuscript project, and this contains underneath it all sorts of projects associated with manuscripts, and when we click on any one of those will themselves contain data sets. Sometimes one data set, sometimes more. The project I just clicked on is a project that we've called HIV Demo. The data set for the HIV Demo project is available on our GitHub site, so that if you install PhyloBook locally, you can grab this data set and test on it. One of the things that you'll see when you click on a project is it will tell you what project you're in. You're in the HIV Demo project, and that the HIV Demo project in this case just contains one data set or one phylogenetic tree. If we open up that phylogenetic tree by clicking on full, now we get a fuller view of that phylogenetic tree, and if it's a long tree, you'll have to scroll up and down to see the whole tree within PhyloBook. Within PhyloBook, there are a number of tools that can allow us to annotate that phylogenetic tree. This link allows us to show those annotation tools. And as I mentioned on the previous slide a little bit ago, that we can automatically or semi-automatically assign lineages by using some external algorithm. In this case, I used a k-meteoids algorithm, and when I select 5 clusters in k-meteoids, it labels my phylogenetic tree now. It says that these guys in red belong to one lineage, the guys in green belong to another lineage with the green triangles, the ones with the black triangles yet another lineage, and the ones with the yellow triangles yet another lineage. Now these triangles just display what this algorithm has produced. At this point in time, we have not yet assigned each of these sequences to a lineage, but we can do that by hitting this apply to labels. And as soon as we do that, what we're going to see is now each one of the sequences has a box around it telling you what lineage it belongs to. Another thing you might have noticed is that around the outside of this data set, there's now a red bar. That red bar indicates that changes that I have made have not yet been saved. So I'm going to hit the save button, and that's going to save this data into PhyloBook's data system. The other thing you'll notice is that now, in addition to having this plot, we have a new plot that showed up. So let me discuss these two plots. The first plot here has the sequences on the x-axis, that's the sequence position, and on the y-axis are the various sequences. So every line in this plot corresponds to a sequence in the phylogenetic tree. The first sequence is the reference sequence, and so on that line there are no markings. because the reference sequence has no changes relative to itself, and that's what this plot shows, are changes relative to the reference sequence. The changes have a key up here, so if it's an ApoBec mutation, which is a specific type of mutation in HIV sequences that are of interest, it's marked with a bright magenta dot. If it's an A, C, G, or T, it's marked in a different color relative to whatever the reference was. If there's a G to A mutation, it's in a specific color, and gaps are shown in gray. So this plot shows the variation within this data set relative to the reference sequence. This plot only appeared once I had assigned lineages, and it shows variation in these sequences relative to the assigned lineages. One of the things you might notice is that there are colors in here that correspond to the colors that we've used for the lineages. So there's a gold and a black and a blue color. There's also a lot that is highlighted gray. We can see a green color over here. The gray color indicates that there's variation in these positions relative to all of the sequence. There's some variation, but there are multiple sequences that match here. You can see that this position corresponds to this big gap. So all of these sequences and most of these sequences have a lot of matches to each other that don't match this lineage that corresponds to the gap. We'll come back to that discussion in just a minute. But over here I can hit hide multiple matches, and now this match plot gets cleaned up. It doesn't show the ones in gray, the ones where sequences are similar to each other but very relative to some other sequence in the data set. So these two plots can be used to help us identify sequences that belong to the same lineage. Now, one of the things I'm going to immediately recognize is this sequence, which has a large gap in it, probably should be grouped with the lineage that's labeled red. It's just that my algorithm for predicting what that sequence belongs to put it in its own bin because it had a big gap. So I can reassign that to the red lineage by drawing a box around it, just holding down my mouse and clicking, and I will click on red, and now this sequence has been assigned to the red lineage, and again you see we have a red box around our data set. If I go up and save that, now this blue lineage will disappear, and we'll see that these sequences match the red lineage best. These sequences match the green lineage where there's variation between the green and the red. And then there are sequences that are all black. And then there are sequences that look like they're likely recombinants, because these sequences have some features that are common to this black lineage down here, and some sequences that are common to this red lineage. This sequence here also looks like it's a recombinant. It has some features that are common to the black and some features that are common to the red. So I'm going to assign this sequence right here as a recombinant. And the way that we do that, our convention is the recombinants get assigned into this yellow box. Now I'm going to go through and I'm going to draw boxes again just by holding down my mouse and going around all of these sequences. All of these sequences in this grouping are recombinants. And we can see that by seeing that they share features in common with various signed lineages. And then these two down here that I missed with my previous box are also recombinants. So now I've assigned these guys to recombinants. I'm going to hit the save button again. And again, my match plot will update a little bit. And if I look carefully down here, this sequence, this demo 38 underscore 1, this one, if we zoom in over here on the match plot, what we'll see is that it contains some features that are similar to the gold guys and some features that are similar to the ones in black. So that one is also a recombinant. It's a recombinant between sequences from this lineage and sequences from that lineage. So I'm going to go ahead and assign this sequence as a recombinant. And then I'm going to go up here and hit save. So now I'm fairly happy with my assignments. as shown by these boxes, and what I'm going to want to do is extract these lineages. So if I hit this button here, assign lineage names and extract to file, I get a warning. And the warning that I get in this particular case has to do with how we've set things up for our use, which is we have a convention. And our convention is that when there are multiple lineages, we name the lineages by multiple lineage 1, multiple lineage 2, lineage 3, etc. And we color them so that the most abundant one is red, the next most abundant one is blue, the next most abundant one is green, etc. Now, in my initial lineage assignments, the blue one had a single member that had a big gap. I've since put that guy into the recombinants down here in the yellow group, which is named recombinant, and there's nothing in the blue group, so this is telling me that my lineages are not assigned in our standard way. So I can hit this button that says recolor by lineages, and now those have been recolored the way that we would like them to be colored, and I can hit the assign lineage names button, and those guys have all been properly assigned. So now we can see, oh, here's the second most abundant lineage, here's the first most abundant lineage, etc. All the yellow ones are recombinants. This guy down here in the bottom is the fourth most abundant lineage. So if I go back up and hit this button, just hitting the assign lineage, now I can download all of these lineages. They're going to go to a file, and I can save that file wherever I want to. We'll save it on desktop 1, and because I've saved it once before, it saves with version parentheses 1, etc. Alright, so now I've shown you how to assign lineages, how to export the lineages and save that data. I'm going to show you a few other features that are available for our use within PhyloBook. If you mouse over to this edge right here, one of the things you see is the mouse changes. And what that does is it allows us to drag vertical lines up and down across this plot. And so if I was concerned, is this mutation right here in this sequence common to other sequences within the plot? What I can do is I can drag this bar on top of that mutation. I can zoom in and say, does that same mutation occur elsewhere in my data set? And it looks like the same position here. It's perhaps the same mutation as we're seeing in other sequences within the data set. Similarly, there's a bar up here that I can drag down, and this will allow me to more easily correlate data within a match plot, mismatch, or a match plot with a very specific sequence because it highlights all the way across. Alright, so there are some other tools that are available to us that aren't particularly applicable to this data set, so I'm going to stop for a second and then we're going to pull up a different data set. So now I'm going to go back into my projects. I'm going to go into my manuscript data set project, and I have in there a data set underneath this project called Washington SARS-CoV-2 spike. This is also a data set that's available for download with the software. It's a data set that I produced by identifying 10 sequences per month of SARS-CoV-2 spike proteins that were obtained from virus in the state of Washington over time. And so if we zoom in on this, what we'll see is we have some GenBank IDs, and then I've collected sequences and I've named them based on the year and month in which those sequences were found. So 2021, January, February, March is 3. February 2021, there's a 2021, February 2021, January 2020, the 11th month, etc. And one of the things you'll notice, and not a big surprise, is since the virus evolved over time, there are dates that are in clades that appeared later. These are Omicron sequences right here. These are sequences that look very similar to the original Wuhan sequence in this particular data set. And because I've named my sequences with fields that are separated by underscores, I can use a tool within PhyloBook to color the sequence names by some of those features. So now when I hit on the annotation tools to show them, one of the things that you'll see is this field is available to me, and this field is available to me to be used to color these sequences. Now, this field, month, would be available to me if it wasn't for there are more than 10 flavors. We've limited PhyloBook to show things that are in these delimited by underscore fields as long as they're less than 10 different flavors within a given field. You can use them to color. It turns out that when you have more than 10 different things you want to indicate, it's really hard to have 11 or 12 or unique colors that are easy enough to see. And so month is not available to us, but year is. So if I click on year, what I can do is I can assign anything in this group. In this case, this one doesn't have a year. This is the original Wuhan reference sequence. But I'm going to assign that and anything in 2020, this blue color. I'm going to assign anything from 2021, the magenta color, and I'm going to assign anything from 2022, we'll make that light blue. And so now if I color the sequences, you'll see that the sequence names have changed color, the font colors have changed, and you can also see there's a key up here. If it had a 1 or a 2020, it's blue. If it's a 2021, it's magenta, and if it's 2022, it's light blue. And so you'll see as we go down, here are all the sequences from 2022. These are mostly Omicron sequences. There's one that appeared early, but you can see it's 2021, but it's in December of 2021. So those sequences now get labeled in a way that makes sense according to the year. Now, depending on what you had of interest, you could name your sequences with different names or fields that correspond to geographical location, fields that correspond to tissues, fields that even correspond to some other feature of the virus that you're particularly interested in. But this is just a way to allow you to annotate the tree based on fields that are encoded within the virus, or within the name of the sequence, I should say. So we could hit save again, and that will save those annotations now as I have created them. Okay, last thing I want to show you is just a little bit more about what it looks like when you have multiple trees. So if I go back to projects, within projects I can have sub-projects, and within those I may have multiple trees. So here are trees that were generated from a project where we got multiple copies, and so now you can see that it's possible for us to have those across multiple groups. This one tells me that there are 10 of 10 trees. There are other projects I have that will have even more trees than that. So we can see we have, in this case, 132 trees within this project. When I have lots of trees, they're only displayed in groups of 10. And when they're displayed in groups of 10, I have navigation tools up here that will allow me to jump to the next group of 10. So now you'll see we're on 11 through 20. To the next group of 10 will put me at 21 through 30, etc. Or I can go to a specific grouping, and now I'm in 101 through 110, where that grouping has been selected on by name. I can jump all the way to the end, the last couple of trees in my 132, or all the way back to the beginning. So this should feel very familiar to you compared to other types of navigation tools. There's also some admin buttons up here which will allow me to do things like assign users and groups. These admin tools will only appear to you if you are an admin within the system. Alright, so that concludes right now my initial Introduction to PhyloBook and how to use it. In a separate video, I'm going to talk about using these admin tools, installing PhyloBook, and then in another video, we'll talk about how to process your data to bring it into PhyloBook.